everybody. Welcome to another episode of our podcast, The Living Algorithm. So, Sushant, hi. How are you doing today?
Good morning. Doing very well.
Great. So, folks, today we are diving into a provocative and controversial idea that fungi and yeast, or more broadly, intracellular fungal pathogens, may play a central role in causing or driving cancer through what's called the cell suppression theory, right? So, Sushant, my first question to you is what exactly is the cell suppression theory and how does it differ from the standard somatic mutation theory and metabolic theory of cancer?
Okay, so to understand this, we have to understand the Warburg effect, which was Otto Warburg told in 1929 that, even in the presence of oxygen, the mitochondria is not able to transform energy ATP, right? And, because of that, the cell resort to the pre-symbiotic era of, using fermentation or model to generate energy, burning glucose. At that time, it was only glucose what Otto Warburg found that. And this was all because, the mitochondria had become dysfunctional. It could not even produce ATP, even the presence of oxygen, right? And the cell to survive resort to this fermentation model. And this was the model because when they ferment, they produce lactic acid, and lactic acid is like a wall for cancer cells to prevent from any attack, right? And for even to drive their growth, right? So that was and this was earlier refuted by a lot of oncologists and healthcare practitioners saying that no, there still is oxygen and it can be oxidative phosphorylation can be reinstated, but essentially it was found that later found in which was also reinforced by Professor Thomas Seyfried, who came to our talk last year, right? So essentially he clearly reinforced that mitochondria are using oxygen to produce ROS, reactive oxygen species, that is damaging your DNA, RNA, and protein, and the mutations and the DNA damage what you have is just an outcome of this defect in the mitochondria, right? And again, but this was never explained that what causes this damage to the mitochondria, because mitochondria is the powerhouse of our cell, not only from a energy transformation perspective, but it is a distributed network. It can sense environment and send signals to other mitochondria, even to nucleus, to drive epigenetic changes, to drive metabolic reprogramming, right? And to move to a new allostatic state. And that is so for instance, when there is an some insult, mitochondria can release cytokines and other signals that could communicate with your nucleus to turn off specific genes, right? That drives epigenetic modifications, that drive metabolic changes. So all these are, your nucleus receive instruction from a mitochondria to make certain epigenetic modifications, right? So essentially, even in a paper, in a recent paper, it was found that around 70% of the epigenetic changes are driven by a mitochondria, right? And now then mitochondria have their own DNA and they could shed their own DNA. It is used as a communication model to communicate you know, uh, with nucleus, and it also indicates an onset of a mitochondrial disease, which includes cancer as well, right? So this is the basic groundwork of what we are discussing today, right? And again, there were no clear explanation of what causes defects in the mitochondria. Now, what we have also found through our data and through our customers, that there are a lot of you know bacterial, fungal, and viral pathogens that could overgrow or become virulence, and express virulence genes, and that could trigger a lot of inflammation, which could damage your mitochondria, which could lead to production of reactive oxygen species, right? So these could be your gut bacteria, your oral bacteria, your gut microbiome, your fungal fungi, and fungi are dimorphic, so they can act as fungi or they can act as yeast as well, right? So they confuse actually cells and immune cells basically, right? So they can take different forms. They can form biofilms to attack your cells, right? So that is what we are going to discuss today about cell suppression theory.
That's right. That's a very interesting take. I mean, no one would have thought that these organisms, fungi, yeast, they could actually do anything. But still, I am really curious to know that what role does chronic inflammation plays in allowing these opportunistic fungi to establish an intracellular infection as per this particular model?
Okay. So what happens is that, when there is an overgrowth of your fungi or yeast, probably you are consuming you have certain dietary choices, maybe consuming too much of sugar, ultra processed foods, or maybe, I'm not sure, but I suspect that this yeast fermented protein could also feed these fungi, right? And they can overgrow. When they overgrow, they dominate the ecosystem, right? And they could
Hmm.
suppress other other bacteria, or they could even collaborate with viruses, right? Okay. So when they can collaborate with viruses, they can overgrow, and when they grow, they could trigger infection. They could produce toxins, that could
Hmm.
actually, they try to enter specific cells, and they try this infection and toxins could damage your mitochondria. So as a result, there is a cell danger response, right? So what happens is that in case of cell danger response, cell decide that I am not going to use oxidative phosphorylation, so I'm going to use oxygen to kill this pathogen, right? To prevent this, this onslaught of toxins and infection, right? But during this process, when you block oxphos, which is a way of producing ATP via respiration, right? You go back to the pre-symbiotic era of fermentation of glucose, which produces lactic acid, right? So essentially, this is a normal process, but if the infection is sustained, or this pathogen already done a damage to your mitochondria, then the fermentation model is sustained, and this is what allows cancer cells to grow. They start fermenting glucose and glutamine as well. So they produce lactic acid and succinic acid that let them grow. And then again, when there's a lot of this inflammation, these injury happening at different parts of cell, immune cells gets into act. They go, and then probably because immune cells are programmed to actually enter exit different cells, and so these immune cells can actually fuse with these cancer cells, and they can become rogue, and that's how we could this lead to metastatic cancer, right?
So, my next question to you is that, this particular theory, it frames cancer cells as suppressed rather than being wrong or purely mutant. What does that mean, that practically for how we think about the tumor microenvironment or immune evasion and metastasis? I mean, how will it impact the
Okay,
whole
okay.
tumor microenvironment?
So what happens is that, when there's a chronic inflammation because of bad diet, bad lifestyle choices, which lead to growth of pathogens, right? And when there's a lot of these overgrowth of pathogens, right? This chronic inflammation creates an environment for these pathogens, including fungal pathogen, viral pathogen, bacterial pathogens, to actually dominate. And this facilitates the process of infection and production of toxins, right? So when there's inflammation, and this leads to weakening of immune system, right? So these bacteria, these fungi can form biofilms, right? So as to escape the from your immune cells, and they try to invade the cells. Now, as I mentioned about cell danger response, right? Because of cell danger response, you know, this actually creates an issue. Yeah. So we were talking about, the cell suppression part. So when there's a chronic inflammation, this chronic inflammation allows, weakening of immune system, and these pathogen form biofilms, and when they form biofilms, they could easily, escape this immune cell tolerance, right? And when they pass on this, they could easily dominate, evade the cell. And when we talk of this cell danger response, so cell resort to this fermentation model where it start producing lactic acid. And also when there's chronic inflammation, there's a release of iron, and there's an iron overload, right? And again, these lactic acid and iron is super fuel for these pathogens, fungal pathogens. They start growing. And again, these lactic acid and iron is actually, they actually suppress immune cells within the vicinity of chronic inflammation. So immune cells are meant to tame down inflammation, but because of this, these byproducts, these fermentation byproducts and this iron overload suppresses immune system, and this allows the fungal to grow, right, and to dominate the cells, and they kill the mitochondria. And then again, since these fungi love fats, right, so they can easily incorporate in your cell membrane, because we know cell membrane, the more there's a lipid bilayer, and around one third of this consists of a lot of these omega-6 omega-3 fats, right? So essentially they love fats, and they actually overtake your cell membrane, and that make cell membrane dysfunctional, right? So that is how they dominate, and they suppress the cells, and then they try to deplete the nutrients of the cell, and then cell may resort to more fermentation model, to both fermenting glucose and glutamine, right, to actually drive the growth, right?
Yeah, I understand that. Now, speaking of cancer patients now, they are mostly immunocompromised and prone to opportunistic fungal infections. So, is there a way that we distinguish between the fungi driving cancer from the cancer allowing fungi overgrowth? So, is there a way to distinguish all of that?
Yeah, so there are ways, probably, the current healthcare system do not do that, because they're still, obsessed with somatic mutation theory. So you have to understand there are 10 hallmarks of cancer. So probably we can
Hmm,
discuss in detail about them in a later part. But again, these 10
hmm,
hallmarks of cancer is what defines cancer. And your somatic
hmm.
mutation theory only defines two of those 10 hallmarks. So somatic mutation
So,
theory is not consistent with these 10 hallmarks of cancer. So what it means that
hmm,
for all the different forms of cancer, these are the hallmarks that represent. So some of them could be, inhibition of apoptosis, it could be,
hmm,
angiogenesis, pro-angiogenesis environment, right? Defect in the mitochondria, right? So I'm just naming few, right? So essentially, this bacterial theory of suppressing cells and causing defect in the mitochondria explains all 10 hallmarks of cancer, right? So ideally, these healthcare practitioners still are focused on identifying gene mutation and killing them with drugs, right? And again, again, the effectiveness of drug depends upon the mitochondria and our microbiome, which we have discussed last during our last conversation, right? So ideally, there are models where one like those we are using. So we measure fungal overgrowth via oral bacteria or gut bacteria, right? So we can measure fungal overgrowth, we can measure yeast overgrowth, we can measure bacterial toxins, right? And also another thing I want to mention is something called, there's a process in which the immune cells prefer killing viruses rather than fungi, right? So when there's a fungal overgrowth, immune cells would be busy in killing viruses, and that's what I could suspect. There could be a coordinated relationship between virus and fungi, so that fungi overgrow, right? So again, that is another way why fungi would grow and would suppress your cells, right? Dominate your cellular ecosystem, right?
hmm. Now, speaking of the treatment part, uh, there's this interesting thing that, some drugs used in metabolic or repurposed approaches have an antifungal activity. So, do you think that is a coincidence or does it supports the complete model?
Yeah, it does supports. There are anti-parasitic medications. There are certain other medications as well, right? Like fenbendazole and all those. Those are
Yes.
anti-parasitic, antifungal drugs, right?
Hmm.
And there's also a research which clearly mentioned that there was a case of pancreatic cancer where it was caused by fungal overgrowth, and the oncologist prescribed antifungal medications that actually helped
Hmm.
uh, overcome cancer, right? So essentially, there is a clearly a role of anti-parasitic drugs, antifungal drugs in cancer, right? Specifically when there's a fungal overgrowth. So first you have to identify that, right? And again, these drugs could really help in overcoming cancer, right? But I have to also mention another thing which is very important from cancer perspective. We have discussed about nitric oxide as well in oral health
Yes,
in
yes,
our previous
yes.
conversation. Now, so
Time
what
again we
fun?
have.
Yeah. So
We,
now
yeah,
fungi also does one more thing, So fungi, what it does is it sequester, arginine, right? Because arginine is required to produce nitric oxide. So we all know that endothelial cells could use arginine to produce, nitric oxide and L-citrulline, right? So again, arginine, it sequesters arginine so that nitric oxide cannot be produced because nitric oxide is required to increase blood flow and, drive oxygen delivery. So it could prevent cancer. So if you have enough nitric oxide production, it could deliver oxygen, it can produce more it could stimulate mitochondrial biogenesis, it could stimulate PGC1 alpha, which could stimulate mitochondrial biogenesis, it could, inhibit, kill bacteria. So essentially, nitric oxide has a very important role to play in preventing cancer. And since we all know that, 99% of the people who have cancer have oral infections, right? So again, your oral microbiome, people who have oral infections have functional loss of nitric oxide production. Via both pathways, via both endothelial pathway and oral microbial pathway, right? So essentially, this fungi could also sequester or could actually, trap, your arginine so that your body doesn't produce nitric oxide. So that could be another reason for fungi to overgrow, right?
that's true. So, what do you think that, what should patients and clinicians take away today? I mean, they should be focusing on research priority, complementary strategies for cancer, or caution against unproven antifungal regimens. What do you think should be the priority for, people who are in this cancer space, be it patients or clinicians or oncologists?
So, they should now start reading papers, and they should even implement commercial solutions around microbiome, both gut and oral microbiome, and we that we are the only one in Asia using measuring oral microbiome functions and the metabolic activity with the oral microbiome, right? So essentially, they have to do away with this theory of random genetic mutation, even germline mutation or secondary risk factor, as we discussed, right? So if you have a
Mm,
functional healthy mitochondria, these mutations do not will not cause cancer, right? There may be 5%, you know, inborn error metabolism mutations, genetic mutations that may cause cancer, right? So essentially, leaving that, you cannot use the same gene sequencing to give pharmaceutical drugs to treat cancer. So you have to understand the role of microbiome because your effectiveness of drugs also depends on your microbiome. So you have to understand your gut microbiome, oral microbiome functions. I'm not saying of DNA here. And there's some stupid companies who are using microbial DNA and making a lot of obnoxious claims, right? But they are you have to understand what they produce and how they interact with the mitochondria, right? That can help us understand the fungal overgrowth and yeast overgrowth, right? And that could also lead to maybe inclusion of antifungal and anti-parasitic drugs also a part of treatment protocol, right? Because first is you have to identify whether fungi is actually being using biofilms to grow. You have to measure fungal overgrowth pathways, which we do measure yeast overgrowth pathway, and also look at the kind of foods you're consuming. So another thing I forgot to mention about, when there's a chronic inflammation, there's a release of histamine, right? And histamine further lowers your L-arginine levels, and histamine actually could is also an outcome of mast cell activation syndrome, which lowers your arginine level. And histamine could also lead to blood sugar dysregulation, which also is a precursor for cancer, right? So when you have high blood sugar level, it could damage your DNA enzyme, DNA, RNA, and enzymes, right? So it could glycate your cells, so it could make your mitochondria dysfunctional, right? So even a chronic inflammation, whatever sources it may be, it could trigger production of histamine, which is generally are a defense mechanism to allow immune cells and nutrients to get inside the cell. But this actually overproduction of histamine could lead to blood sugar dysregulation, which could, even trigger your increase your risk for cancer. Right. So ideally, again, another point I want to mention, because last week I was delivering a talk on cancer among certain government public sector bank employees, and what I found that a lot of these people, the oncologists are telling that you should not consume fat, you should consume sugar. Right. This is the most you know flawed and wrong thing I have heard, because your dietary fat does not make you fat, and you need fat to strengthen your cell membrane. So people who have cancer may start losing weight, and their cancer cells may start converting the protein, the amino acid in their muscles to produce sugar, and that's why doctors say start consuming sugar, because there's something called cancer cachexia, which actually makes feeds, so the doctors will say, okay, you are losing weight, start consuming sugar, donuts, and all these complex carbs. You are actually feeding your cancer cells, right? So essentially you have to get into ketosis. You have to, because in most of the cancer except prostate cancer, they are, the fats are stored in lipid droplets. Again, they cannot be used by cancer cells. So there are lipid droplets in the cytoplasm which cannot be used by cancer cells. So again, these feeds the healthy cells, and these starve the cancer cells, right? So there are a lot of misconceptions about cancer treatment in the market right now. Yeah.
I get it, Shant, and I think today's discussions have opened another avenue for many people who are into therapeutics to reconsider their treatments, right? So, since we've come to the conclusion, would you like to give a message for all our listeners?
Yeah. So for doctors and healthcare practitioners, please start including microbiome and
Hmm.
mitochondria in your diagnosis protocols. Do not,
Hmm.
just fall for this flawed somatic mutation theory. Please, it does not explain more than two hallmarks of cancer, right? So you ask
Hmm.
yourself
Hmm.
what you are doing, right? You should combine metabolic therapy with your standard drug therapy. Also, consider parasitic antiparasitic drugs, right? There are a lot of research around it, so you should read the research around it. And for people, fix your oral health. Oral health is the starting point of all forms of cancer, right? It is also
Hmm.
a starting point for type 2 diabetes, cardiovascular disease, chronic kidney disease, IBD, and a number of other health conditions, right? So please fix your oral health. Do not go for RCT, which is root canal treatment. It is going to disrupt blood flow. It is going to increase your risk for cancer, right? Right? Maybe
Hmm.
some
Hmm.
dentists may not agree, but that's a no, that's a proven science, because there are each teeth is connected to a specific organ, and there's a flow of electrons and current and blood flow to that specific organ. When there's an RCT, you damage the nerve root.
Hmm.
There's no blood flow, there's no voltage, there's no electricity, no current, there's low oxygen environment, and cancer cells grow, right? That is how cancer cells
That's
grow,
correct.
right?
That's right.
so
That's right.
so so these are the key factors. You want to reduce the risk of cancer, please fix your oral health, right? For doctors, please start focusing on microbiome, right?
Thank you, Sushant. That was a very insightful discussion today. You opened up a lot of other things that one could consider for cancer treatments and therapies, right? So, thank you for sharing this whole, your research-backed information. So, folks, thank you for listening to our Living Algorithm podcast, and it will be really helpful if you share it with your peers and family and friends, so that they can actually get the basics about how the cancers and their treatments work, right? So thank you, Susha.
Thank
And
you.
folks, we will be back with another episode next week. Thank you.
Thank you.